Welcome everyone to the BrainSpace Initiative. It's my pleasure to introduce Danilo Stock. So he's a medical doctor and a computer scientist, and he has a background in both systems neuroscience and machine learning. He currently is an associate professor at McGill's Faculty of Medicine, and he's the Canada CIFAR AI Chair at MILA, Quebec, Artificial Intelligence Institute in Montreal, Canada. So he's been doing a lot of work with machine learning, and trying to move towards using neuroimaging and genomic data to get us towards single subject prediction, which is a very challenging problem. So that will be the topic of his talk today. And so I will leave it to him from here. Thanks, Danilo, for joining us. I'm looking forward to your talk.
Thanks so much for inviting me. You can hear me okay?
Yep, perfect.
Yes, okay. So how much time should I occupy here? 30, 40 minutes?
Yeah, that would work.
Roughly?
That works well, yeah.
Okay. All right, so yeah, I mean, interrupt me anytime if you have questions along the way. That's perfectly fine. So yeah, Danilo Stock. My team is both at McGill's Faculty of Medicine, where we have the Montreal Neurological Institute, or the Neuro, which is known for neuroscience, as well as MILA Quebec AI Institute, which is more known for machine learning and deep learning. And so our work is really sitting on top of both these different institutions, and that really very much explains what exactly we're doing. So we try to revisit classic questions in neuroscience and medicine to tailor new machine learning paradigms to revisit those classic questions, and then translate those insights back into the neuroscience and medicine domains. So I'll give a talk where we'll dive through three bigger themes of how exactly we try to make machine learning work in the neurosciences. So the photo slide. So I talked to and compensated by different companies. And so why is this particularly relevant now? I think we are pretty much going through a transition phase in the neurosciences at this point. And one way to motivate it is 10 years ago, so when I finished medical school, there was a time when we didn't really have large-scale data sets. We didn't really have population-scale data sets. And that very much started to change, maybe initiated by large-scale funding initiatives such as the Human Brain Project in Europe, which was more than $1 billion, $1 million of funding for dozens of labs. And then the US followed up with the Brain Initiative. Israel, Japan, and other countries had similar large-scale initiatives to support neuroscience research. So that is why we potentially have more PhD students in the neurosciences than perhaps in any other domain. This is what the figure on the left here shows. So, and one of the consequences that this has is that we have started to build much larger resources and data sets. They're more multimodal, more people, larger sample sizes, more features than we had at any other point in history. So now the question is, of course, how can we go and analyze those large data sets? So this is where machine learning comes in. It's fair to say that we still experience an exponential development in machine learning where since 2010, 2012, roughly speaking, we realized that convolutional neural nets at the time really started working, and that keeps on working with its current instantiation, large language models. And so the point is that machine learning and deep learning typically need very large data sets to work in the first place. So we didn't really have sufficiently large data sets in the neurosciences. to really exploit a lot of the machine learning algorithms that we have now, or the ones that we had 10 years ago. So this is how these two parallel developments actually are super relevant to each other, especially also for the clinical goals. That's like the last aspect here, personalized medicine. To do personalized medicine, we need both. We need those algorithmic solutions, but we also need massive data sets to be able to really get anywhere close to making statements about a single particular individual. So from my biased perspective, what's the thing that is really at the core of personalized medicine? I would say it's the transition from group case control comparisons to statements at the single subject level. So if you think about some of the sharpest tools in the toolbox that biomedicine has, for example, the randomized clinical trials, they have the form of one big group having a certain treatment, another group having another or no treatment, and then contrasting these groups at an aggregate level. So it's instructive to think about and realize that most of the clinical guidelines, a lot of the clinical knowledge that is the basis for the guidelines of how medical doctors treat patients in the hospitals, this is very much based on kinds of studies that compare groups of people. So the problem is that a certain medication, let's say, may work on average if you compare those two groups. However, even if a drug is beneficial on average, it may have no effects for certain people in those groups, or it may have even adverse effects for certain subsets of those groups. So, and this is for me kind of the main difference in trying to make concrete steps towards personalized medicine. So we have these three mega events that are coming together. And this is why it's a particularly fun time to be playing around with machine learning models in large biomedical data sets. So what is the form that this takes? Let's take the example of psychiatry. I could give the same example about neurology, but psychiatry is a particularly prominent example because we have what the machine learning people would call label noise. So the prediction targets, the group distinctions, the labels, the disease categories, we know they stand on very shaky grounds. And machine learning and big data, from my biased perspective, are probably some of the best ways to escape impasses that some of these research streams may have reached. So also psychiatry, because if you look at how this works in the clinics, a lot of the medication and treatments that are administered to psychiatric patients, they don't usually work at the first attempt. So it takes weeks to identify whether a certain drug for depression, for schizophrenia, bipolar disorder, others, whether they actually work for a certain person. And then people try another drug, wait another couple of weeks. As you can imagine, this is very costly for the individual, for the health system, for society. So, of course, we would like to be able to better select a particular treatment and match them better to a particular individual based on their profile, their demographics, and their genetic blueprint. So how could you do this? The first step in psychiatry, since we know that our disease categories don't actually carve out distinct entities in nature, would be to just potentially just mix them up. You could just build large data sets with all sorts of diagnostic labels and comorbidities, because we're not really sure about the boundaries between these disease groups anyways. So that's what you see in A here. And then B, ideally, we would like to have data sets with a lot of very different kinds of information. Massive genetic profiling, so the most common is still common variant genetics. We went through the time of epigenetics, and more recently single cell genomics, but you also want a massive deep profiling, so what some people may call phenomics. So all sorts of indicators, quantifiers that allow us to index and monitor activities in everyday life, aspects that we did not typically measure in traditional in-lab experimental studies. So, and of course, brain imaging and brain-derived information, such as magnetic resonance imaging, it's still a prominent one because it's easy to deploy this at scale. So we just combine all this information and then put machine learning models against this to let them do what they are good at, searching the needle in a haystack or finding the intrinsic dimensions of variation in the data, which is what you see here in 3 axes in a lower dimensional space. So the basic assumption from a technical perspective here is, are there lower dimensional descriptions that we can apply that may be completely different from the disease categories that we started with, but based on this wealth of data and multimodal data put all together, can we find a description system that actually allows us, and we're panel C here now, that actually allows us to carve out some biologically defined subgroups. So that is why we have these homogeneous colors here in each of those subgroups. And I think that this is an unavoidable milestone on our path towards precision medicine. We will need to go through the stage where we have these big data-derived subgroups to do something that the precision medicine people try to do, which would be, for example, diagnosing patients much earlier than we can do now, or as I already alluded to, picking treatments better, personalized for the person in front of a clinician, or as well try to anticipate the trajectories that a certain patient may take. And so this is roughly on a conceptual level, the workflow. And as you can see, you have the machine learning component and you have also the big data component that work together. So one of my favorite points that I've been making tirelessly over the last 5+ years is that the kinds of tools that the machine learning and deep learning communities use, they are of a very different nature than the ones that 20th century biomedicine has been using. So there's a lot of ways in which you can pit them against each other. But first off, from a historical perspective, a lot of the tools that we still use in biomedicine are here in the blue part of this timeline. It's interesting to consider that most of the techniques that a lot of research papers, studies use still today, they're actually invented before Second World War, before people had calculators broadly available, and before people had desktop computers that were broadly available. So that emerged, p-values, null hypothesis testing, ANOVA, all these techniques, they were emerging from a time where we didn't have big data sets. So potentially no area of human activity actually had big data sets, but it also includes biomedicine. So in red now, in the second part of the 20th century, we saw the emergence of what we today call machine learning, with tools such as cross-validation procedures, structured regularization techniques such as retrogression, so early precursors of today's machine learning, such as resampling techniques, like the bootstrap. Around 2000 only, we invented random forest that can exploit higher order nonlinearity. and so on and so forth. But all these techniques, they're very different in character because they require more data, as I already said, but they also require much more compute and storage capacity. But the most important point for me is that the kinds of research questions that we can ask with machine learning tools, they are not necessarily perfectly overlapping with the kinds of research goals that classical statistics in blue has. So what I'm saying is you cannot necessarily take a certain research paradigm or goal that we had hunted with traditional statistics and then just exchange the tool and have the same exact research goals as before. I personally believe that the interpretation and the kinds of questions that you can ask and answer with machine learning, they're partly distinct. Okay, so that takes me to, after this conceptual introduction, to the first theme of machine learning enabled research for neuroscience, for biomedicine. And that would be Nerv's paper from now 7 years ago, which is about atlasing. So, you know, even experts in the field, computational neuroanatomists, they don't usually have a clear answer for the question, how many brain regions do actually exist in the human brain? So that's a seemingly simple question, but it's a surprisingly difficult topic if you really want a precise answer. So why don't we just learn the atlases from the data themselves? And that's the approach we have taken here as an instance of transfer learning. So I'm not going to try to give a rigorous technical definition of transfer learning. For our purposes, the way I usually explain it is you want to extract some form of structured knowledge from a larger, typically more general purpose data set to then take the derived quantitative model or algorithm, carry it over to a more narrow, more specific data set. This could be a low sample clinical data set, for example, to derive value, derive insight based on the structured knowledge that you have extracted in the general purpose data set. So you want to carry over insight from a large data set to a small data set to do something that you couldn't have done otherwise. Okay, so the way we did it here is we recombined the, at the time, largest data sets in the imaging neuroscience community that existed, CAMCAN, Human Connectome Project, and so on. And we trained, or actually we tailored a deep learning model to learn a representation, a compressed representation of the most relevant pockets of variation in these functional brain scans. And we did this by predicting all sorts of experimental tasks. So here we only have task-based fMRI data. So the point is that we don't actually want to classify these outcomes, but we just use it as a pretext, if you will, to train the model to identify a simplified representation of which parts of the brain, which sets of voxels actually matter the most for people interested in task-related distinctions in the brain. So, and that's what you see on the right-hand side here, these parcellations. So the point is now, you can take these parcellations, go to a smaller boutique data set, just apply this parcellation that we have derived in a data-driven way, and that simplifies the analyses and the data science workflow a lot, and you can do all sorts of analyses just based on these image-derived atlases that you couldn't have done otherwise. Another quick example is based on the intuition that, you know, when a radiologist gets trained, that's maybe somewhere in the mid-20s, that individual has already experienced the external world for more than 2 decades. So what I'm saying is the specific knowledge in the human brain that is related to distinguishing X-ray, CT, MRI-based information for the purpose of making diagnoses. Well, this stands on the shoulders of several decades of trained visual cortex circuits to decode the external environment. And so why don't we use computer vision models, train them on some of the largest existing data sets for videos and images, which is YouTube, to then try to improve the prediction accuracies in brain scans. So that's what we tried to do in this proof of principle study here, we published 2 years ago. So we demonstrated that you can interpret a video from YouTube just as a series of 2D slices, similar to MRI scans in the brain. You can use the pre-trained model, the YouTube pre-trained model, to then go to what, from a machine learning perspective, is a small data set, to the UK Biobank imaging, to then show that you can outperform MRI-based phenotype prediction, such as sex differences, age prediction from MRI, which is a popular task, or also phenotypes such as IQ. You can see gains in the out-of-sample prediction accuracy if you start off a model that has actually been trained on information that has nothing to do with MRI scans, namely YouTube. So that's another example of transfer learning. My favorite and last example for the transfer learning theme is based on text. So I'm sure you heard about that there's always more interest in text data. We live in the age of large language models now. And so that's going to be the first original article, hopefully from our lab in this realm, and that's applied to autism. So let me explain this in a few sentences. The problem, the challenge of diagnosing autism is that the prediction accuracies that we see based on high throughput technologies like genetics and MRI, they're not actually that high. So my personal reading of the literature is, if you really look at technically rigorous, accurately conducted data science workflows, you cannot usually do better than 80% accuracy based on the whole genome profile or based on a whole brain MRI, fMRI, or structural MRI. So you could argue that it's kind of surprising that we can't classify autism better. So we thought we can just go several steps back and instead machine learn the clinical intuition of medical doctors themselves. Because trained psychiatrists are, for a fact, pretty good at diagnosing people with autism. So why don't we go and machine learn that? So we use state-of-the-art large language models, pre-trained on millions of articles before. So in this case, it was tens of thousands of psychology and medicine-related articles, just think PubMed. And we then go to a set of 4,000 reports, handwritten documents from medical doctors, and we deploy our pre-trained model on those. So we do see prediction accuracies that actually are higher than most of the previous studies based on MRI or based on genetics. But that's not even the most interesting. The most interesting is we found ways to actually decode the internals, the viscerals of this large language model. Many of you will know that the explainability problem is a big one. However, for this particular application, we found ways to, for example, say which particular sentences in these reports are how similar to known DSM criteria, although we didn't even measure those in these people. So we can make more accurate classifications of autism just based on clinician text reports. And we did find strategies to actually unpack these large language models to gain insights about what is it exactly, what are the facets, the aspects of subjective experience that across dozens and hundreds of clinicians are actually consistently valuable to identify who is a suspected cause of autism, a suspected case of autism, and who is an actual case of autism in this data set. Okay, so I hope I convinced you that transfer learning is actually an important one. So diversity-aware modeling is the second of two themes. So what's diversity-aware modeling? So if you Google this, you're not going to find a lot of information. That's a term that we try to introduce. My favorite way to motivate this is this plot here. What you see is on the x-axis is the sample sizes of particular studies, and on the y-axis you see what out-of-sample prediction accuracies they report. So if you know, if you did fit a regression line on this plot, to me it's pretty clear that this line would actually be showing a downward slope. So the higher the sample sizes that we look at, the worse the prediction accuracies. And that's the case for all sorts of different diseases. You see them on the right here: autism, depression, psychosis, Alzheimer's. So from another perspective, the more we go towards representative general population data sets that are not the result of very strict inclusion and exclusion criteria, the more we see a decay in out-of-sample prediction accuracy. So the big question is, of course, why is that? Is this for technical reasons? Is it for biological reasons? So we try to make some progress in this admittedly very challenging question. So here's another piece of evidence that witnesses these challenges. So two of the large data sets at the time, a Human Connectome Project and the ABCD cohort on the right, across dozens of phenotype predictions that neuroscientists and clinicians typically care about. Here on the x-axis, we show that if you take a predictive model that was trained on the entire cohort, the out-of-sample prediction performance, that's what you see here, it actually differs as a function of the subgroup. So African Americans in blue, actually in those subgroups we see much worse prediction performances, oftentimes below chance level or at chance level. Whereas in white Americans in green, this is where the models are really working. So there's a big difference here in red between subgroups for predictive models trained on the entire cohorts. So even worse, let me just summarize this at a very high level here. What we published 2 years ago here in this PLOS Biology paper is we don't actually have good ways to correct for even known subgroups. So for example, this can be side effects, it can be ethnicity, it could be socioeconomic status. So what you see on the right here is that the more diverse our training cohorts, the more diverse our out-of-sample cohorts, we see a decay in the prediction accuracies. And this is the second line on the right here, linear deconfounding strategies that we apply on a daily basis, they are not able to mitigate this influence of diversity. So in newer, more recently embraced techniques, such as, for example, COMBIT, that's bottom right, that doesn't help either. So the problem is we cannot apply our established incumbent deconfounding techniques to correct for even known subgroups. So it's really worth investigating further, in my opinion. Here we look into the predictive rules that these models actually extract. So if you try to predict disease categories from MRI data, it turns out that if you change the diversity of your training cohorts deliberately as an experimental variable, you see that the predictive principles that these AI models latch onto, they are actually quite diverging. So that's what you see here in these correlations. There are different regimes of predictive patterns that we actually reliably identify. And so the point is that as a function of the demographic constitution or composition of your training samples, that actually drives a huge amount of what you even observe as spatial patterns in the brain for a particular disease. So, and it's even more complicated. That's the last slide for the second theme, diversity-aware modeling. Because we have spatial specificity in the brain for how much brain behavior relationships depend on training sample diversity. So it turns out that that's what you see here on this slide. The higher you go up in the neural processing hierarchy, the closer you get to the higher association cortex, the more brittle the predictive rules, the predictive and AI models actually extract. So it's especially the default mode network, dorsal attention network, and frontal parietal networks that suffer the most from diversity-related background variation in terms of how well we can reliably identify what parts of the brain are actually related to a particular disease category. So one speculation that we have why this is in our team that the higher association cortex is more live wired, quote unquote. That's known. So what do I mean? For example, in adolescence, we know very well that the higher circuits are more malleable for a longer time throughout life. So that's maybe because these circuits keep learning to a larger degree deeper into adolescence and maybe even early adulthood. But the point is that that also makes them more susceptible, these parts of the brain, for differences in the environment, in a certain culture, in a certain geography. And that's perhaps what we are witnessing here. Okay, so I hope I convinced you, summarizing the second theme of using machine learning for precision medicine and single subject prediction. We will probably have to deal with background variation that is driving a lot of the results that we see in all kinds of studies. And I would go as far as saying that background variation in all kinds of data sets is perhaps even overwhelming a lot of the target effects that we are hunting, because brain behavior, cognition, disease kinds of associations tend to have small effects and tend to be brittle. Okay, so last theme that's going to be quicker, and that's this question of when should we actually use a certain tool for a certain job? So I'm just going to give a caricature here, which is you know, a new fancy AI tool is hyping on Twitter last week, and then the idea is, hey, this is a popular technique. Why don't we use it in neuroscience and biomedicine? So I think that per se is not necessarily a good strategy. Biomedical data is very particular. A lot of our data, common brain genetics, brain images, they are not very much like natural images or other benchmark data sets that the machine learning community has. And so this really motivates the question, when should a particular class of models be used for a particular goal in neuroscience or biomedicine? And so the concrete answer from my subjective perspective would be, why don't we run more empirical simulation studies? So that's what we did here. We worked in the 10,000 UK Biobank imaging sample. That was the largest at the time. So that's 5 years back now. And then we tried different classes of machine learning tools, like 2 generations ago, maybe in red, linear predictive models, then 3 deep learning tools, so seeing green, and then blue deep learning tools, which should be even more powerful, identify not only nonlinear patterns like the green ones, but actually deep nonlinear patterns. And so the inductive bias should be, if we have nonlinear structure in these data, then the blue models should be able to outperform the green and the red ones, which is what you see in the schematic on panel C. So we compared the scaling behavior of these classes of models in general benchmark data sets of machine learning. That's what you see on the left-hand side. The MNIST data set, which is handwritten digits, 10 class classification problem, as well as Amazon Fashion, which is
Daniel, are you online?
Here. Deep learning really does better, whereas in structural MRI, that's not exactly what we observe. So we actually see a collapse of these modeling regimes. Questions here for this one?
No, I just
No?
lost you for a second. We will keep the
Oh.
question for the end. Thank you.
Okay. Yeah, so I'm almost done. I'm roughly in the time frame that I mentioned at the beginning. Okay, so the big surprise to us was that at that time, with this sample size, pretty much one of the largest imaging data sets, clean imaging data sets, uniformly pre-processed data sets I'm talking here, not retrospective data collections from completely different studies from completely different parts of the brain, the planet. What we observed at that time is that we couldn't really distinguish the scaling behavior, so the empirical sample complexity, as the machine learning community says, between those deep learning models in blue, nonlinear models in green, and linear predictive models in red. So with also 10-class classification problem. So we then thought, okay, is it maybe because we are not looking at the right part of these brain scans? Is there maybe a feature selection step that's missing? So we tried all sorts of ways, particular slices, particular searching and screening methods. And I can say that across all these analyses, we saw the same collapsing of these learning curves. So there wasn't really a reason based on these data that nonlinear tools necessarily do better than linear ones. So then the big questions, of course, and we were asked this a lot during the paper revision process, what could be the reason that we actually observed that? So that's very hard to know. One potential culprit, I alluded to it, is noise. So what kind of noise? Well, we can measure random noise or we can simulate random noise. And that's exactly what we did. We took the general purpose machine learning benchmark data sets, the MNIST, and we repeated our analyses, deliberately adding always more random noise. And that's what you see across these panels here, with the final observation that as we increase more noise, we see always more collapse between the high capacity predictive models and the linear predictive models. And so that is a hint, that's not a proof that MRI kind of data and potentially other kinds of biomedical data, they just by their nature may have more noise. And we know from theoretical results that it can be difficult to escape the linear modeling regime in the high noise setting. So that means even if you have nonlinear structure in your data, you for fact do have nonlinear structure in data. For example, gene interaction. We know there's nonlinear effects. However, it's a different question whether you can actually reliably extract that. So here's the good news now. We repeated this analysis in 40,000 UK biomic imaging subjects and even introduced extrapolations to what we may get if we had 1 million subjects with imaging data, structural, functional, and diffusion MRI data. That's what you see here for a whole bunch of phenotypes. And it seems that if we have more than 10,000 subjects, 40,000 plus, there is indicators that we can still improve the prediction accuracies. But if you look at the y-axis, these increases in prediction accuracy may be modest. And so, yes, we can probably train always more complex models with always more training data in MRI. However, the question is whether there isn't maybe a diminishing returns or the effort in compute storage, training of lab members, and so on is actually justifying the increase in the out-of-sample prediction accuracy. So this last study of this kind was published in Cell Reports, if you're interested. Okay, and that takes me to my final slide, where I'm just going to say that I think there's a lot of value in building research, insights, but also rigorous machine learning platforms, which isn't trivial, for empirically justified single subject prediction. So why am I saying empirical? Well, because going back to the initial example of psychiatry, what I like to argue is that, maybe we don't need the mechanism. So that's a kind of a statement that is somewhat unpopular, what people typically don't want to hear. I like to argue that to improve single subject prediction, to predict, does a certain symptom improve? Does a certain drug potentially work better? Does a certain person actually have this disease trajectory or this other one? I personally am not convinced that we desperately need to understand disease mechanisms at the molecular level. I'm not saying that's not useful, but I think it just is at a different time scale. And we can make way more progress, do way more good, if we just apply machine learning to big data in a way that is fast, cost-effective, and pragmatic. And in the parallel stream of research, we can still go and try to hunt down this molecular mechanism. But I think this is just going to take way more time. I thank my collaborators and these financial institutions for the support, and I'm happy to take any questions.
Great, thank you.
42 minutes.
Good timing, good timing. So I guess I'll start. I have a question or two. It was nice to see the follow-up to the Schultz paper on the larger data set. I was wondering if the results you were showing were based on the 3D CNN, or those were just using the slice-based of the 2D models.
So we also included 3D information.
Okay. So as you know, we looked at that as well, and we found with even small numbers of subjects a benefit when you use 3D CNNs. And we thought that was maybe due to we found something kind of in your paper that kind of explained it. I thought, I don't know if you've looked into that further, and if you tried that fix and did it help?
I didn't, because the first author left my lab, and so we didn't
okay.
really follow up on this.
Let's chat.
Because I didn't really have control over this
there was
scientist.
Yeah, there was, just for that particular model, we found in your code, I think the target and predicted variables were swapped for the 3D model. So we can point you to that and see if that changes results at all or whatever. We found it did make a big difference. So just a small thing.
Okay.
But I agree with you. I think in principle that deep learning models are not a silver bullet. And I think it really does depend. And sometimes very simple models eat the pants off of more complex models. It really depends. Sergey Pliss here has been looking at that a lot as well, kind of showing that you can do pretty well in prediction just with some fairly simple, even they're maybe, they're deep models, but they're shallow, they're shallow deep models. Okay.
Makes perfect sense.
Yeah. There's no, this is nothing new, right? Modeling, there's no perfect approach to any of this stuff, right? So, before I'll let other people ask questions too, but when you looked at obviously your decay with number of samples, right? Obviously there's obviously there's the well-known, I think you referred to this as a technical reason, which is like the winner's curse, right? So you have sampling variability, and you can show in simulations that you get this cone, and it kind of, it decreases, and so it should go down based on that. But did you differentiate that from other reasons, and how important are those other reasons? I was just curious because you did differentiate a little bit in your talk, and I was wondering what you had done there.
Yes, that's a very good question. So the honest answer is I've been writing grants for this for 4 years and then never got funded. I just find
we can all relate.
It's very difficult to convince the NIH kind of reviewer pool or the equivalent institutions in Canada that this is an important topic. So I completely agree. We would like to know to what extent do we have random noise or structured noise? And in the structured noise, to what extent is it biological or environment or technical, like scanner differences and so
Yeah.
on? I think if we could quantify this, at least to some extent, that would be a very valuable contribution.
Yeah. Yeah. And like, is the winner's curse more of a curse for certain complexities of data, for example, or diversity
Yeah.
or whatever, all of those things.
I agree. I agree. So what
yeah.
we could, let me say it this way. We didn't get funding for exactly the studies that you outline. However, we have tried to make progress in, as you saw, diversity aware modeling. So the turn that we took in my lab is that we try to do horizontal innovation by translating and tailoring modeling solutions that allow you to integrate known sources of, we call it major population stratification, into the modeling. So instead of just looking at a brain behavior relationship, like how can you predict age from different parts of the brain as a function of major sources of population stratification? So that's what we have been working on a lot, and that's going to be the next papers that come out. We have a Nature Reviews Neuroscience paper under review that we just submitted the revision that's just going to be fully dedicated to this topic. And we have a Nature Methods paper that's actually cited here at the bottom that has the conceptual primer. So this is how we try to make some progress there.
Cool. Okay. There's a question about simulation. So it's really, what is the, let me pull it up here, chat. Okay. So synthetically generated data, for training, we've been looking at this, again, Sergey's group in particular, that there are models like segmentation models. You can almost train those with no data. You can just randomly rotate the brains and you don't need big data to train deep learning models for those. Other cases where we're trying to study huge, populations, and maybe we do need large samples, and then there's in between. So just curious how you think about that, what your thoughts are, the role of simulation, the role of data augmentation, the role of getting more data, all of that stuff. Yeah.
I mean, I'll need to ask, what kind of simulation exactly? Do you mean a synthetic data generation as a
just
form
broadly,
of data augmentation?
Yeah, just broadly speaking, right? So
yeah, okay. I would say, so as a very simple statement, I'm surprised that the common variant genetics community hasn't come up with something like that. And maybe I missed something, maybe it's more complicated than I think, but I don't understand why they didn't really go forcefully into this direction. In MRI as well, I mean, for example, from a data science perspective, if we talk MRI, I think the last 20 years of ICA, including your work, Vincent, and many other people, Oxford, and so on and so forth, it's pretty obvious that there's a very informative low-dimensional space in MRI images. So it's hard to deny, whether we call it resting state networks or something else. And that's what a lot of MRI studies these days are based on, and what they implicitly accept. So as a first approximation, to me, it seems completely legitimate. You could just fit a PCA model, any ICA, anything like that, autoencoder, if you will, and you can just do combinations of these latent spaces, even conditioning on disease categories or something like that, and generate millions of examples of realistic, plausible MRI images. So to me, that makes perfect sense. But I haven't really seen that. But that's not because I may just have missed that, because I don't really read this particular pocket of literature closely.
Oh yeah,
However, I would have expected that
yeah,
people do this more.
yeah. No, I was thinking like what the one extreme example is the you can take a set of labels for a segmented brain for segmenting a brain, and then simply rotate, shift, move, that kind of stuff to train a model that the SynthSeg approach, right? It works pretty well on clinical grade MRI scans, just using simulation to train it. So it's kind of an extreme example. Anyway, I don't want to take up too much more time, but I also like your comment was controversial that maybe we aren't interested in mechanism. So that's
and let me just make my main point because I forgot it. Look, if you go to medical school, you get the impression that the gold standard way of doing things is a single scientist has a genius idea, mouse model, then showing proof of concept with an agent acting on the mechanism was identified, then scaling it to humans, RCTs, and then it's like a widely administered drug. And one of my favorite comments at this point is that never happened in psychiatry, to my knowledge. So this pathway of how drugs are even validated, that's absolutely not what led to the drugs you actually use. So that's a fair point. So this idea that you come with this ingenious human who has this brilliant idea, and then it goes all the way through to a drug that's sold to millions, that's not actually true in this form, at least not in psychiatry. So empirically, you can just very clearly argue that we should have parallel streams of research.
Yeah.
Yep.
We just haven't found the right genius person yet. No, I don't know.
Yeah, maybe.
I think the only thing, this is different from your point, but the only thing I would mention is you kind of you mentioned the word explainability. I think visualization is another term I like to use, which is I think there needs to be more visualization. Yeah, maybe not mechanism, but more visualization, because I see so many
high dimensional spaces.
Yeah, but I mean, visualize a part of it, right? I see so many papers that have no visualizing information in them, and I almost at this point just move on at that, because I'm like, and the only reason I say that is because in my experience, when you visualize, you find mistakes and you find problems.
Yeah, I couldn't agree more. So
and if you haven't shown me that, and we being all of us, if we haven't shown each other that we've done that due diligence, why should I trust the results, right? So
exactly. Yeah, so I totally agree. I mean, that's there's no controversy there. I mean, that's in fact one of the big sections in this, what hopefully will be Nature with Neuroscience. Let's see. We specifically say, actually, one of the most useful applications for machine learning tools may actually be in visualization. That's exactly what we say, because maybe the subgroup categories that we have in mind, they are not actually the ones that drive the variation in the data. So that's one point. And the other empirical point is, I would say if one of my team members says a certain pipeline doesn't work or the predictive model doesn't work, 80 or 90% of the time, it's somehow related to what goes into the model, that we haven't carefully enough printed and visualized all the inputs to the model. That's, from my limited experience, most often the reason why something doesn't work. So this is exactly confirming what you say.
Two quick questions. One, in the plots you show, I'm not sure which plots are referring to, but this is Ellie's question. You showed degradation of method performance on MNIST data. Was that added with the added noise, image noise or label noise?
Image noise.
Image noise. Okay.
So we blurred the actual handwritten digit information.
Okay. And then the other question is, what do you think, Morgan is asking, what do you think of Alto Neuroscience's approach to drug discovery? Are you familiar with that? I'm not sure.
Yes, I know Ahmed Etkin. We actually talked at the Society of Biological Psychiatry meeting where we also met last time.
Okay.
I don't know exactly how he does it from a technical perspective.
Hmm.
So we don't usually talk about that. As far as I understand, he also does a form of subtyping or subgrouping. But I don't know how exactly he does that.
Okay.
So I can't really speak to this. I can only
Okay.
say there are all the indicators of success.
Okay. Great.
So far.
Great. Oh,
Mhm.
and then one more I think we'll ask from Oliver for the transfer learning example that uses physician reports. What is the percentage
Mhm.
of the correct diagnosis? And does your classification accuracy outperform it?
Yeah, that's this plot here. So that's unfortunately not the plot that's not our final plot because we just got the last tranche of data, but it's hovering around the 80% here. So we get at around 80% out of sample classification performance, just based on text data, so beggar words kind of representations. That answers the question? Or what's a differently nuanced question?
Yeah, physician's diagnosis. Sorry.
The physician's
yeah.
diagnosis. Okay, that's this question: is the eye better than the clinician?
Does it beat the doctor?
That's not the point. Okay, I understand the question. So, well, that's what I call the inference prediction dilemma. Yes, I emphasized accuracy here. That's maybe misleading. We effectively could also run the study if we are barely above chance. We could. I just mentioned this in passing. Most of the time when we do machine learning in our lab, we just want to know that we have a stable predictive model. Once we have that, we go and try everything to unpack what exactly this model learned. So the main contribution here on this paper we're going to submit in a few weeks is going to be that we can actually unpack the large language model. We're not trying to do some benchmarking of prediction accuracies. Yeah.
Is there any bias in the prediction? This is a question from Mo, because physician diagnosis is based on the physician report.
Bias. Well, so it's even more subtle because I mentioned this only briefly. The actual groups that we start with is actually, are the people suspected to have autism or do they actually have autism? So the thing is, we are not actually comparing against a healthy group. It's actually more difficult, if you will, because all the children that are in the study, every single data point is an individual who was suspected to potentially have autism as judged by a mental health professional. And so whatever the biases there are, what I can also say is the clinical institutions where we got this from here in Quebec, Canada, they see almost one autism patient per day. So they have a huge amount of clinical experience and they're completely specialized to autism. And since they're specialized, they really draw widely they have all sorts of kind of strata of the population. So if that's the question of bias, then I would say it's fairly representative.
Yeah, great. Well, we're at 2:00 now, so I think we'll probably go ahead and close, but great talk. This is really interesting. And for the BrainSpace initiative, we have a Slack channel that will open up and we can save the questions, and there may be some follow-up discussions so people can kind of get an opportunity to interact with you, and we'll also have the talk shared as well there.